Okay, thank you. Hi, I'm Matthew Madany. I am a grid applications developer at the Department of Neuroscience at UC San Diego, and I've been using the power of Parsl to help me solve a variety of microscopy problems that I've described here as hierarchical because they come from a wide range of problems that you need a more stronger and sophisticated parallel computing setup to kind of deal with them on your feet actively. So I'm going to describe that problem here. So we developed these workflows that analyze tissues mostly from brain in electron microscopy and 3D electron microscopy. So that immediately presents one of the biggest challenges in the computational workflows we have to develop because we deal with multidimensional data as an input. Usually data doesn't become highly dimensional until the intermediate stages in computational workflow design, but we have it as an input, and then we're most interested in developing a sequence or series of AIs, and AIs are also highly dimensional. So when you have these input volumes, which can scale exponentially because of the XYZ axis, you can very quickly end up with what we call teravoxel data, where you have to design something that scales to the problem. And so you can have an AI developer who can maybe solve a sub-problem or a sub-volume or a subsample using a few hundred lines of code. But then when it comes to the teravoxel scaling, it takes thousands of lines of code and a parallel computing expert. However, the parallel computing expert could maybe do it in a few hundred lines of code. But then maybe the machine learning experiment isn't going to scale. And so I've been trying to kind of tether together a few Python libraries to make this into something that happens easily, scalably, and can deal with this problem, which I describe at the bottom, which is where you have these 3D input volumes, but the AIs themselves are also 3D. So they need to be able, they're basically pulling out and plugging back in 3D windows, little chunks of data. They're being extracted, factored into tensors, factored into batches, factored into whatever the machine learning problem is. So whether there's ground truth data, whether there's another microscopy mode being leveraged, and then it has to blend that back into the original volume, and it has to keep GPUs flush. Whether there's multiple GPUs, they have varying degrees of VRAM. There's been a lot of work in CPUs and giving them, terabytes of RAM, but you just don't have that kind of architectures with GPUs. So making programs efficient and having workers that actually sit there and expose these chunks of data to whatever your node and GPU parallelization scheme is a very big challenge. And so for that reason, a lot of AIs just don't get built because of the scalability. But I've been able to build them because I've leveraged a couple of libraries, which I'll describe here, to combine them and make them into something where you can really pass these abstractions, just configurations, and then define a really quick and easy scalable voxel processor for AI. So the first and probably most important one is Parsl. So that one is your parallel execution engine. And with training and inference, there's a pre-process to training, there's a post-process to training. With inference, there's a pre-process to inference and post-process for inference. Sometimes it's serial, sometimes it's sequential. But the one thing it always has in common is that it's multidimensional data. It comes in as 3D data. And then with AIs, you have batches, you have channels, you have depths, you have multiple data sets. And so it's not uncommon in my application design to have like 6th or 7th dimensional data sets flying around. And handling the overhead of that is very difficult, which is why I use this as our library, which is a data concurrency scheme where you define these files. Here these N5 files and these data set names, and it takes the dimensionality of basically your NumPy array and decodes that into individual files that are based on a chunking scheme that you define, and then that gives you a data set which efficiently exchanges data between RAM and disk, such that you can essentially trust it regardless of whether it's 10 terabytes in whatever node or storage system you're using to feed into your AI when it's training or inferring. And so I just give a little framework for what I'm doing. I'm starting with PyTorch. I'm actually wrapping Parsl inside of a PyTorch data loader. And there's kind of a beautiful irony to mention that the deep learning frameworks like TensorFlow and PyTorch at their core are also data flow parallelization engines. They always talk about it's a machine learning library and how great that is, but when you look at the code, the majority of it is actually handling data flow parallelization, because the machine learning frameworks have to move tensors in batches through multiple models that could be at various stages of completion. But Parsl is there to basically handle that parallelization framework at more of a global level beyond that GPU. And yeah, using this, at the end of the day, I accomplish what most programmers want, which is, instead of making tens of thousands of lines of code defining workers, how many CPU cores, what happens if you're on a file system that can handle millions of files versus thousands of files, what happens if you're on a file system that can has more rapid I/O versus slower I/O, and how do you design applications that just deal with all of that. But with this framework, I've been able to get it down to like just a few hundred lines of code, just regardless of the input of data, regardless of what cluster you're running on. You just pass it a few configuration options to Parsl and ZAR, and it handles the data and the CPU scaling such that you're not worried about your GPU scaling. So this is one of the first applications I built that was really, I felt like I just couldn't build it without this. This is electron microscopy super resolution. So this is where I use generative adversarial networks to actually artificially interpolate resolution that I just don't have because of the limitations of the microscopy. So these are 3D volumes, and one of the things about pretty much all of three-dimensional imaging is that usually two of the cardinal axes, the X and the Y, regardless of whether it's even electron or fluorescence, whether you're using photons, electrons, or X-rays, you tend to get more resolution on X and Y than you do on Z. And that has been a huge limitation for actually solving the structures that you're interested in analyzing in these data sets. And so using these generative adversarial networks, I've been artificially interpolating that resolution to actually make these microscopy volumes look more like they should do in a high resolution domain. I'll show a video of that. So in this video, the bottom row is the original volumes as they were acquired raw from the microscope, and then the top row is going to be their artificially enhanced, or their z-axis has been given an extra slice in order to help with visualizing the structures and giving you a microscopy output that otherwise would have required a microscope that had to be very delicately engineered and would have been a lot more expensive, and there would have been trade-offs with throughput and quality, and that's a whole other presentation. And so what I hope you appreciate from this video is just the smoothness of this upper row versus the bottom row, which the bottom is the raw and the upper is these generative interpolated. But one of the things I'm trying to sell here is that it's not just that I created an AI that I normally wouldn't have, or that wouldn't have scaled. It's that I'm able to kind of on my feet just create a bunch of them that have different functionalities. So this one in the upper left is a multi-label voxel classifier. So this one I train off of ground truth data that I annotate to label all of the structures in these microscopy volumes. So in these color overlays, there's labels. These pink ones are synapses. These teal ones are neurotransmitter. The green is the boundaries of the neurons. And so that's kind of a scalable microscopy application that I wouldn't have considered building in the first place if I didn't have this kind of data processing scheme that I could wrap efficiently. This one on the right is content-aware image restoration. This is where we have variations in standard operating protocols and it's biology, so it's messy, and so it's very common to get an image that you just weren't expecting or didn't raise to your standards because of who knows what. So we can train algorithms to actually understand what the problems were and fix those and resharpen the image. This is the super resolution one I just presented. There's also microscopy domain translation that I've been working on, which actually takes two mutually exclusive sets of microscopy and maps the resolution to the lower resolution mode. I'll show a video of that one. So this volume on the left is serial block phase scanning electron microscopy. So this is where you have a block of tissue and a knife cuts the top of it and then it images the block. You can get usually pretty high XY resolution, but pretty low Z resolution. The one on the right is the same serial block phase electron microscopy volume, but it's been translated to the tomography domain, which is a much more higher resolution isotropic microscopy domain where you get equal resolutions across X, Y, and Z axes. So you'll see the features and these overlays. So you see this is a myelinated axon right here. You'll see that the features are translated to appear as they would in the higher resolution domain. So when I play this video, you see this stepwise chunking scheme, and here you see the smooth microscopy you get with the isotropic domain, despite the fact that they're disjoint data sets. And you can imagine that this is where the scaling problem really comes in, because what if you had a terabyte data set and your Z resolution is 10 times less than your XY resolution? Well, you could fix that with the AI, or at least let it give you these artificially enhanced ones, but you'd end up with 10 times more data, as well as intermediate data that is very complex, highly dimensional, and requires kind of this Parsl data scheme I've been using. And then I'll just take you through my last examples, which is just, this is the whole point. So we can do reconstructions, these very complex reconstructions of neurons with there's intertwining axons, dendrites, and all of the interesting components that make up the subcellular structures that define the synapses and all of the energy components of the cell. And you know, when neurons fire and have their action potentials, there's all of these structures beyond the neurons that are actually involved in modulating and making these signals synchronous in the brain, and we understand virtually nothing about them because of how difficult it is to hierarchically solve all of the microscopy problems such that you can do these reconstructions I'm displaying in front of you. So this pink is like a forest of axons, this blue is the dendrites intertwining with them, and here I kind of have a zoom cut out of the same one where I'm showing you that we also annotate these structures inside of these neurons, which are involved in signal modulation and are also very important for studying neurodegenerative diseases. This is an example of these composable workflows. So it's really dynamic in that I'm making these workflows that have all of these GPU stages to kind of converge on these final reconstructions. Usually you just have your CPU stages that build up to your GPU stage, and then you have your CPU stages after that. I really kind of sit down and I'm like, how do I get these 5 or 6 or 7 GPU AIs to really work in a serial or sequential batch? And then here in the bottom right, I just have these analysis of what we call the neuropil, which is the part of the brain where you have intertwining axons, dendrites, representations from hundreds or millions of neurons, where we understand very little about how that just structurally contains itself and defines itself in any kind of micrometer scaled volume of the brain. And so I'm just plotting some univariate analyses of like different brain areas and how the synapses kind of change structurally across different brain areas, or the types of synapses and so on. This is kind of my higher level orchestration resource dashboard, where I'm just showing that I did my job. I'm keeping these GPUs flush, and I'm showing execution. Here's the GPU dashboard. This top row is how many GPUs have been assigned to me. And then this bottom row, this row right here, is their utilizations. So I'm just showing that I did my job, because at the end of the day, we just really want these GPUs to actually have their utilities above 50, 60, 70, 80, 90%. So a lot of people build these AI applications, but then when they put them on the cluster, when they connect them to the data, because of those data schemes, because of those parallelization schemes, there just aren't enough CPUs or data buses that are sufficiently flush enough to keep that GPU at like 80, 90%. But with this scheme, I am able to do that. So I'm actually staging this and orchestrating it globally with Kubernetes on the Pacific Research Pipeline. And so I have heterogeneous resource definitions where I could be on a node that has who knows what kind of GPU. What is happening to my screen?
The hearts are Dan's way of signaling that we're at time, I think. I don't know how to do it.
Oh, okay.
How do you do that?
And here's some institutions.
Awesome. Thank you very much, Matthew. So we've got time maybe for one quick question before we move on to Ian. Anyone have a question? So I guess my question, I know a little bit, I just found out about this work actually, but Matthew, the plan is that you're going to share this in some way as a a library that other people can use this? Because I guess you're combining a bunch of sort of general purpose tools into a scalable framework for doing this type of analysis, right?
Yeah, I have plans to release it as software, but I probably need some help from your group.
Well,
if I
more than
wanted to
happy
do
to
that.
help. I think
Yeah.
this will be, I think, very useful, usable for people that do any sort of microscopy, even imaging people at APS, etc.